Oh, that's interesting. How does this thing work? Ah. Testing, testing. Hello, hello, hello. Good morning. Can you all get to where we stopped in the notes? Maybe you'll get there before me. We had some very simple little examples that we can talk about. I think this is where we stopped, where we were talking about charged amino acids. And I should also give you a chance to ask any questions. Any questions from last time? Anything that is still unclear from anything that we talked about? So this first slide is actually the next three slides are pretty much quick review. So let's just go over it pretty quick here. What happens to the charge on an amino acid if the pH changes? Let's be specific. What happens to the charge on an acidic amino acid, say if we decrease the pH? Or increase the pH. You can talk about either one of them, right? So acidic amino acids, say we're at pH 7. What kind of charge is on an acidic amino acid, like glutamate or aspartate? pH 7? It's what? No net charge? People agree with that? At pH 7, what kind of charge? Come on, guys, help me out. Negative, right? So what about if we increase the pH more, above 7? What's the charge going to be on a negative amino acid? I mean, on an acidic amino acid. What was the pKa for an acidic amino acid? Around 4? Is it going to make any difference if we increase the pH any further? No. Okay. So pH affects the charges on amino acids, right? The negative ones particularly. What about if you had a basic amino acid, basic one that like lysine that has a pKa of 8.3, pH of 7. If we increase the pH, we're not going to probably. Let's talk about decreasing the pH. Does decreasing the pH affect the charge on a basic amino acid like lysine? What kind of charge are we talking about? Positive, right? Is it going to change if we decrease the pH? No, nothing's going to change that because the pKa is so dang high, right? Okay. So if you charge, I think we've answered that. What about if you're within a protein and you change the pH? So acidic, again, our best example because it's the most physiologic example, right? Basic, we said, are probably going to maintain that positive charge almost at every pH that you can think of. But acidic, it's going to change based on where you are in the body, right? When would aspartate have no net charge, as the lady up here said? What's our rule? Well, let's revisit the rule so that we have this written down so that we're really clear about it. I said something like protons dissociate when the pH rises above the pKa. So that means they come off when you're above the pKa. What happens when you're below the pKa? Somebody speak out because it's a big crowd and it's... What happens when we go below the pKa for aspartate or glutamate? You guys really are not with me, or is this just so simple that you just can't imagine that I'm asking it? What is the... Okay, we're at pH 2. Right, right? The pKa is 4. I said protons dissociate when you're above the pKa, so what do you think happens when you're below the pKa? Right, the proton will associate. In aspartate and glutamate's case, what will that do to it in terms of its charge? It neutralizes it, right? It's now a Zwitter ion, essentially. Okay, so what does that do to the structure of a protein when you lower the pH on it? It changes the structure. What exactly, right? It disrupts the structure. I'm going to move us along here because I think we're probably, it's that thing that happens at 10:45, even though it's really, feels like 8 o'clock maybe when it's in this room, maybe with the dank smell that is in this room, which I apologize for on behalf of the university. It's embarrassing. I'm sorry. And I won't tell you, well, I will tell you. I'm going to tell you. So, okay. In 2003, when I began here, this room was so bad, so it's been worse. If you can believe it, it's been worse. The roof was leaking in so many places in the spring melt that, and I taught in the spring, and it was my first semester teaching here, and I'm thinking, what have I gotten myself into, right? There were like 8 garbage cans gathering the kaplunk, 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 you know, as the snow is melting off of the hot roof. Unbelievable. So it has improved a little. Sorry, that was a sidebar. Oh, excuse me. What is the process called that we're talking about? So when you change the pH and in the environment and the protein, amino acids like the acidic ones lose their charge, what happens to that protein? Say it louder, somebody. Out. It denatures, thank you. So it's denaturation, right? And that means it's unfolding, right? Okay. When you talk about that, by the way, be clear what we're talking about. Which kinds of interactions that stabilize proteins are we talking about when we talk about denaturation? Throw them out there. What are the kind of interactions or bonding, you know, that, yes? Ionic interactions, yep. What else? Hydrophobic interactions. What else? Van der Waals. Is there one more that I can think? Well, actually, there are other ones probably too, but sorry? Disulfide linkage. Thank you. I'm sorry. I'm like, whoa, I'm not hearing. Disulfide linkages, yes, they can be broken. Yep. Is it going to change? That's a great question. Again, let's talk about that. So Lane is asking, acidic amino acids have a negative charge at neutral pH, pH 7, right? We said the pKa is what? 4, right? What's our rule? Protons dissociate when pH rises above the pKa. So will it change above the above pH 7 or 8 or 9? I mean, it's not going to get that high. You'd be dead. But anyway, but on the other hand, when you get below the pKa, the protons associate, right? They come back on, and that's a very, very rudimentary way to talk about biochemistry. I know that you know that it's more nuanced than that, but for our purposes, that's good enough. Okay. There's another hydrogen bonding. I'll just throw that out there. Remember, that's the weak kind of things that keep the secondary structures together, beta pleated sheets and alpha helices and all that, right? Okay, so here are some really, you know, here we are moving down the path again of highly simplistic, but food science, right? Right, got to pull in food science. There are people in this room that would be food scientists. I mean, and everyone eats, right? Are there real world examples that we can talk about? Marinating meat, right? What do you marinate with? Anybody cooks out there? No. Okay, so at least to know, yeah. Unless you are a vegetarian, in which case you don't need to know about marinating meat. Although it's helpful if you're a dietitian, because, anyway, what do we marinate in mostly? Or acids of what kind? Not hydrochloric, right? That would be highly effective, but not so palatable, probably. What do we use? Huh? Do you really? In the kitchen, do you use citric acid? I don't, really. Oh, I'm just thinking about cooking, you know. Do you? Lemon, sure. You know what? And that's what's in lemon, isn't it? Right? So I'm thinking biochemically. I'm like, well, citric acid, like a powder, you're going to get. Sorry. Yeah. Lemon juice. Yeah. What else? Yeah, vinegar, acetic acid. That's right. You guys are chemists. I totally am not, you know, I'm thinking food, you're thinking chemical structure, which is good. Okay, so, you know, that kind of thing. I give the example of that. Not that many of you probably do that, but are there other examples, ways that we can denature protein besides adding acid, which is these two examples? What now? Cooking, right. Any of you ever made cream of tomato soup and done it the wrong way? If you add the you know, cold milk to the hot, or actually vice versa, you can curdle the milk in a very quick order. Anyway, so that's generally, you're dropping the pH. What about the last example, more physiologic, is think about what the pH of the stomach is, pH 2, okay? And this is a no-brainer, so I'm not going to belabor this. I think we're getting it, right? Everybody makes sense to you what happens to those proteins. They get denatured, they unfold. What do you think that does to digestive enzymes? Remember, we went over this, so this is all totally a review. What does it do when you drop the pH, the stomach, right? pH 2, hydrochloric acid. Say it louder. Does it activate them? No. Well, only pepsinogen, right? That's true. What does it do to, so that's good. For one thing it does. It activates pepsinogen to pepsin at the stomach, but what does it do to the protein that facilitates digestion? I should have asked it that way. No, we're talking about low pH now in the stomach, pH 2. What does that do to the proteins that facilitates digestion? Or how do you think it impacts the action of digestive enzymes? So just, yeah. Very good. Thank you so much. That is just the review I was hoping for, right? It unfolds them, it denatures them, so it allows the enzymes to actually have access to the primary sequence, so it can start chopping off amino acid residues. Is that clear to everybody, or it's just maybe too early? Yeah?
Did you mean then what happens to the proteins?
Yeah, proteins that you are eating, and when they hit the stomach.
So not the digestive enzymes?
Not the, no. So what happens to proteins that contain amino acids that you're, you know, bringing in, right? They've got aspartate residues and glutamate. Mostly, right, we're talking about the negative ones that are going to destabilize when they become proteinated. Yep. Okay, are there any questions about that? Is that pretty basic and hopefully helpful? All right, now a little more review here. How many amino acids are there? How many? What? 20? Great, 20. Do you remember which ones, how many are considered, and here I've just given you, these are synonyms. Someone was asking me about indispensable versus dispensable. Those are different ways of saying indispensable means essential. It's the same, right? How many? 9, right? And that, you know, you can see various some discussion about histidine, but I think we've, you know, for the most part, people have come to consensus on it. It's 9. And there are different categories. The essential and non-essential, which is the same as saying indispensable and dispensable. And then we've got this conditional category. And, you know, there are a number that are highlighted in the Stepanek text. Ones that I think are relevant I mean, they're relevant depending on what population you're working with. But in growth, for kids, arginine is considered conditionally essential. So conditionally essential in children. There's one that we would call conditionally essential in the ICU patient, somebody who's undergone major trauma, who's undergoing metabolic stress. Anybody have any idea what that might be? It's glutamine. I'm not hearing if anyone's shouting that out. So glutamine is one that is, we would say is conditionally essential. And then preemies, premature infants. Cysteine, which is abbreviated CYS, and glycine. Those are just a few examples. There are probably some other ones. Yeah, in MNT, I'll tell you about how people with kidney failure need extra histidine because they're not able to recycle the limited amount that the kidney deals with. So we'll talk about that. That's another one, histidine and kidney failure. All right. Any questions? Do I need to repeat anything? Which ones are essential? Have you heard of, however you say it? Mnemonics, thank you. I'm always kind of like menominee. I always think of menominee. Menomina. Sorry. Mnemonics. Which one did you learn? I learned Private Tim Hill, which is weird, I guess. I don't know. Anybody have another clever one? Did you learn? Do you remember what they stand for? Whether you had a mnemonic or not, hopefully you remember which ones are essential. What's the P? Phenylalanine. V? Valine, all right. We got 2 T's. Tryptophan? Not tyrosine. It is a T, but it's threonine, right. How about the M? Methionine, fantastic. H? Histidine. I? Isoleucine. And then we got 2 L's. Leucine? Lysine. Great, fantastic. And just, to help you remember some things here, do you remember which one's a branch chain? You want to, no, don't remember? It's all a complete blur, isn't it? Actually, you'll be refreshed after we go through muscles, so we'll talk about these. But anybody? Not a 1? Leucine, yeah, very good. Who said that? Thank you. Is that Austin? Yeah. Okay, leucine. Isoleucine, yeah, that helps you, doesn't it? Valine. I always try to remember it that way. It's branched. Okay. Anyone remember which one has a hydroxyl group of these? This is where if you learned the structures, then it helps a little bit. But which one? Not methionine. Methionine, what's unique about methionine? Sulfur. Yeah, it's a sulfur group. Threonine has an OH group. And that's probably good enough for us at the moment. What makes an essential amino acid essential? We talked about this last time, and I know you know it. So what is the thing that makes them unique? Say it louder, will you? Can't make what part of it? So think about the structure of an amino acid. What'd you say? The side chain, the R group, right? It's the R group that we can't make for whatever reason. Structure can't be synthesized. Oh, and did I put this on your slide? No, okay. So I will just say this is where the controversy around 8 versus 9 stems from. We've accepted that 9 is the number, right? And histidine is the one that was in contention, and it's because it's essential for infants. So if you think about, that's how we establish what is essential and not, is for kids. we break down carnosine at the kidney. We can release histidine, so we can recycle some of it. So for adults, you know, if you went completely histidine free for a while, it would take a while before you got deficient. But I mean, don't worry too much about that. It's just that's a Trivial Pursuit kind of, you know, level. Because someone might ask you, well, what is the deal with histidine? Because you'll see older texts and, you know, newer, different varieties sources that'll talk about that. Okay, did everybody get that? Yell if I change and you need, okay. Okay, a few other little trivial bits about amino acids. You've heard of PKU, right? Phenylketonuria. This is one of several inborn errors of metabolism around amino acid metabolism. And it's a genetically inherited disorder. It involves a defect, in the enzyme that metabolizes phenylalanine, which is phenylalanine monooxygenase. By the way, just for your purposes, whenever you see a yellow box with purple writing, that's an enzyme, and I've done that consistently throughout all of the notes that I do. And I realize you're printing them in black and white. But if you look at them in color, it helps you know which one the enzyme, you probably have gathered by now, you've been around long enough to know what an enzyme kind of looks like. It's got the ASE on the end. But anyway, so it is a problem of the buildup of phenylalanine and phenylpyruvate that cause retardation and you know, delayed development in infants. So the way that we treat it is to provide a phenylalanine restricted specialized formula in infancy to basically reduce and minimize that. So you're supplying all of the essential amino acids except phenylalanine in a formula, and then they get a little bit from natural sources from infant milk, as typically is how it works. In these people, tyrosine becomes essential. That's another example of a conditionally essential amino acid that I didn't tell you. Tyrosine in PKU. Potentially. Although I mean, I don't know how big of an issue it is, but if that was if they were only getting essential amino acids, that would be a problem. Okay, other amino acids that are that you'll see, and you probably are aware of some of these, but 3-methylhistidine, you ever hear about that one? Some amino acids get changed after a protein gets synthesized, so it's called post-translational modification of amino acid residues within the structure of a protein. 3-methylhistidine is one that we used to use as a reflection of muscle mass, lean tissue, and that was for nutritional assessment purposes, but it's not super specific to skeletal muscle. It's present in other, it's in the protein called actin, A-C-T-I-N, actin, and that protein is not just in skeletal muscle, although it's high in skeletal muscle. It's also in, you know, intestine and other tissues. So anyway, but once an amino acid gets modified, it doesn't get unmodified. There's no way to take off that methyl group once you put it there, right? So it's going to be excreted, and typically it shows up in the urine. Then I give you a couple of other important examples: hydroxyproline and hydroxylysine. And I know that the M&T students are going to remember what protein those important amino acids are in. Yes, please. Collagen, thank you so much. Collagen, and the fact that they're hydroxylated, they got an OH group on them. These are highly prevalent in collagen, and I think I have a picture on the next slide, so I'll hold that thought for the moment. What vitamin is required as a cofactor for these hydroxylation reactions to make collagen? Yay, vitamin C. You guys, I'm so happy. No, it's nice. I appreciate that you remember that. And that's important, right? Because wound healing, you think about vitamin C and collagen synthesis. And scurvy, if you're vitamin C deficient, you're bleeding gums. And even if you haven't had MNT, most of us probably remember stories where that has happened, right? And it's not that common, although I did see a case in my practice where somebody developed scurvy. But anyway, Here's a picture, actually, this is kind of what it looks like. It's a triple helix. Collagen is a very strong, you're not going to draw this, don't have to sketch this, it's really, but it's just a very, very strong tensile, strength. That was redundant, but anyway, a lot of force gets put on collagen. And so that triple helix is the hydroxy groups on the proline and lysine. And proline is one of those weird amino acids that kind of causes bending to happen. And so anyway, it allows for this very, very strong helical structure, and then they come together in 3 chains, and you can't break it. Collagen is one of those very, very powerful proteins, which is a good thing that we have it in our skin and other places where we need, wound healing and that kind of thing. Okay, lastly, we got amino acids that are used in neurotransmitter and hormone synthesis, and serotonin, very important, neurotransmitter, melatonin, all coming from tryptophan, tyrosine. We make these, and those again in MNT, you remember what class of hormone those are? What do we call those? Louder. Aaron, did you say? Well, not corticosteroids. That would be cortisol. It does begin with a C, though. Catecholamines, yay. All right, catecholamines. Fight or flight, right? These are the very important hormones. And then I just bring up glutamate acts as a neurotransmitter by itself, and it also gets converted to the transmitter GABA, gamma amino butyric acid. I'm not going to ask you what that stands for, but if you want to write it down, I'll say it again. Gamma amino butyric, butyric acid. And then, for the clinical realm, there's a medication, class of medications for treating depression called monoamine oxidase inhibitors, MAO inhibitors. They're still around. A lot of what we do for depression is serotonin reuptake inhibitors. So we basically keep more serotonin around. It's an excitatory neurotransmitter, right? So that's how we typically, probably more than not, depression is treated with one of those kind of drugs. But MAO inhibitors are still being used, and a lot of people only respond to MAO inhibitors, right? So one of the things that as a dietitian you need to be aware of is that MAO inhibitors require people to follow a diet that would be modified in tyramine. They have to be careful about how much tyramine they get, which is a derivative of tyrosine. It is not tyrosine, you need to know that, but it's a monoamine that comes from it. And this MAO enzyme inactivates amines, okay? So its normal job in the body is to inactivate amines, tyrosine and tyramine, okay? And it's kind of an arrow right here. I've got a slide on the next page, but I'm not sure that I gave it to you, so I have to remind myself if I did. Tyramine would be also leading to the production of norepinephrine and epinephrine. So you need to write that down, okay? Tyramine can yield norepinephrine and epinephrine. All of these are monoamines. So norepinephrine, epinephrine, tyramine, those are all monoamines. And MAO inhibits, it basically deactivates those. And so that's important. If you think about epinephrine and norepinephrine, if you didn't have MAO, this enzyme, you wouldn't be degrading those, and you would be hyped up all the time as you would be if you were running from a bear or something like that, right? So you'd be stressed out, to say the least. And actually what ends up happening when you block this enzyme, I'll show you on the next slide, yeah, can I show you too? Because I'm going to repeat it in the context of, so tyramine, again, and let me just make sure, did you have that last point here? You do, okay, great. So you know it's not the same thing, but it does yield norepinephrine and epinephrine, tyramine, right? Now, MAO inhibitors will degrade tyramine. They incidentally, and I'm not showing it here, it also will degrade norepinephrine and epinephrine. All of these are monoamines. They get acted on by MAO. So if you give a drug that blocks MAO, So MAO inhibitors are going to stop that enzyme. You're going to end up building up your tyramine, and your norepinephrine and epinephrine are then going to, you're going to increase the circulating levels of these important hormones, right? And those can cause hypertensive crisis. You get a really high blood pressure, you get headache, heart palpitations, you feel pretty darn bad, and often need to get into the hospital to actually bring it down. So, we restrict foods that are really high in tyramine. Anybody have any idea what those would be, Lena? Aged cheeses and aged meats. Sauerkraut, all right. Wait, sorry. Soy sauce. Fermented soy foods like tempeh. Smoked meats and cheeses as well. They often are aged too, typically. So they kind of, anyway, but all of those create more tyramine in them. And then red wine, and for some reason Chianti wine, and I have never figured out why that's called out, but red wine in general tends to be higher in tyramine. Any other foods I can't remember that we talked about. So, okay, is that clear? All right, and I'm just going to quickly go through this. So amino acids, they get categorized by their R groups, their size, their chains, hydrophobic, hydrophilic, sulfur containing. I'm going to click on through here. Essentiality, conditionally essential, and then finally by the fate of their carbon skeleton. All right, so we're going to talk about that next. What happens to their carbon skeleton once their amino group gets removed? Because that's what we're going to go to next, is what happens to the amino group. But I'm going to talk about this, I think, right now. That when you say, actually, let me back up just a moment. So when you say glucogenic versus ketogenic, what do you think glucogenic means? And I could say, let me think about this. Yes, I could also say anaplerotic. Remember anaplerosis, anaplerotic? You heard that term before? What is it? So what does that mean when I say glucogenic or anaplerotic? Yeah, it does what to the CCA? Say the first part of. What does it? So what is anaplerosis specific? So you're thinking about the TCA cycle, Christina. So that's great. That's good. You're in the right realm. What is? It's regenerating. It's adding carbon, right? So when you add carbon to intermediates, basically the intermediates that add carbon, we would call them anaplerotic. So you're replenishing. Keeping the TCA cycle going requires adding carbon to it, right? So glucogenic would be, and I'll say most but not all amino acids are glucogenic, meaning their intermediates coming from their carbon skeletons can regenerate or replenish carbon in the TCA cycle. All of them, ketogenic, what do you think that means? I'm sorry, say it louder, will you? So ketogenic, so you got to be thinking about making something. Can make fat, right? So ketogenic means it can be used to make fat. Now, all of them can be used to make fat, okay? All of them can be used to make fat, but not all of them are glucogenic. There is one that we say is uniquely ketogenic, which means it cannot in any way ever be used to make glucose. And that one is leucine. Okay, you might as well write it down here, and then I'll show you on the next slide here. So if you see leucine, I've highlighted in red, again on blue, my apologies, because that's not the easiest to see. Leucine only gives rise to first acetoacetate, which is 4 carbons, and then acetyl-CoA is 2 carbons. It does not show up anywhere in, it's just not. So the other ones, lysine, phenylalanine, tyrosine, these do lead to glucose. leucine is the only one that we would say is uniquely ketogenic. It does not give rise to a net increase in carbon in the TCA cycle. It's not anaplerotic. Okay. So any questions about any of that we've talked about? Yes?
To what detail should we know which amino acids
And I'm trying to remember what I have. I will actually, I would ask you to post the study guide now, if you don't, I mean after class, not now, but immediately following class. For sure leucine, and I think I have picked ones that show up elsewhere in pathways for you to just know and just kind of be aware of because they're so popular that they show up elsewhere. Like oxaloacetate, right, is one. Incidentally, I bet you know this already. What amino acid comes from oxaloacetate? Oxaloacetate is the carbon skeleton of which amino acid? Say it louder. You got to rise above the hum of the crowd. No one remembering? Aspartate, yes. Right? So those are going to be the ones probably I'll ask you to think about. What about pyruvate? Pyruvate, did you know that? I know you've talked all about pyruvate in carbohydrate metabolism, right? It's the carbon skeleton of an amino acid. Which one? Alanine, right? Okay, so those for sure. And if I've highlighted any others, it'll be on the study guide that are most critical. I try not to have you memorize large swaths of information, truly. It may seem like that, but it's really not. Okay, any other questions? Then I think we'll move on to the liver. And please do read the objectives, but I'm going to, just in the interest of time, I'm going to move us along here. So we're back on our roadmap, and now we're going to talk a lot about what happens to the nitrogen here. How does that, and other things following the liver. So first of all, we're going to talk about after a meal. Right? What happens after a meal? Amino acids from the proteins that you've now digested because you have a low pH in the stomach and you've activated pepsin and right, all these other things that went along with that, pancreatic and intestinal enzymes, et cetera. I said the portal vein comes from the gut, right, and delivers a lot of amino acids. And so something to just think about is that, so the liver, you know, that's why we say it's first pass metabolism, is it gets first crack at the nutrients, foodstuffs that are coming in from the diet, right? So those amino acids are coming in the fed state. The concentration of amino acids, you know, is going to be high as it hits the liver. And then we've got this conundrum here, a good bit of them, 60% of them are going to be catabolized almost immediately. So the first question I have is why so many? Why would we just, you know, we take it in and then we just completely, pretty much break it down, not completely, you know, oxidizing all of them, but we catabolize them, we start breaking them apart. Why do we do that? So you can think about what, okay. Okay. So, you know, we could say that, you know, we're going to do a fair amount with those, right? So we can maybe interchange some of them. We're going to talk about how that happens, right? That we're going to use their carbon skeletons. Some of the non-essentials, we can build from essential amino acid carbon and other things. But backing it up to why is it that we have to? Why don't we store them? Okay. Do we store amino acids? Let me ask you this. Do we store them like we do glycogen, you know, carbohydrate or fat, right? We can't really. So we have to use them right away. We're going to talk about the other 40% that we may use, okay? But 60% of them undergo immediate, the beginning processes anyway of we could say catabolism or degradation. We're going to talk about how we can interchange them, and that's an important point. So I'll get back to that. But the main one of the, you know, why so many get immediately catabolized because we don't store them. We have to do something with them, right? We're either going to use them for protein synthesis, or we're going to use them to, interchange carbons, skeletons, so we can create what we need for protein synthesis, et cetera. But how is where we're going to go next. What do you think the first step in the metabolism of an amino acid is for most of the amino acids that we have? Can you think of a reaction that involves amino acids? It really, maybe comes to your mind? Okay, so that's number 2. Number 1, so number 2 is deamination. What's the first one? Transamination, right? By far and away, most of them, the first step in their catabolism is transamination. There are some exceptions. The second would be deamination, where you completely remove the amino group. The where does this happen? Where are most of them getting catabolized? So it's coming in from the portal vein, so guess what? There's a class of liver cell. You heard of it? Called the periportal. Peri, meaning P-E-R-I, around. Periportal. And I'll show you, that's written out many times in this slide set. So periportal hepatocytes is where it's happening. And let me just say this. And it kind of gets to what you were saying too. So the 3 main points about the liver that I want you to jot down. Number 1, it's the primary site for uptake of amino acids after a meal. Primary site for uptake of amino acids after a meal. The vast majority are going to be taken up by the liver in the fed state because the concentration is high getting into the liver. So primary site for uptake of amino acids after a meal. Second, primary site for essential amino acid catabolism except for one group of essential amino acids. So it's the primary site for essential amino acid catabolism except for one category, and that category is the branch chain amino acids. Primary site for essential amino acid catabolism except BCAAs, branch chain amino acids. And the third point is that it's regulating the availability of amino acids to the rest of the body. So that's kind of why the liver is so important. The liver has so many important functions, but that's... Primary regulates availability of amino acids to the rest of the body. Again, it's that first pass metabolism. So it's going to extract a certain amount and then let the rest go through. But a big chunk of them get extracted. And here is a very abbreviated picture of how the intestine and the liver communicate, right? Obviously not built to size, but just so you can remember what the portal vein is doing. It's draining what's in the intestine, lots of things. It's important. This is showing enterohepatic recirculation because we recycle bile salts something like 6 times during a meal. That they go through this. We also recycle B12 this way, so that's just, as a nutrition student, you should have in mind what enterohepatic recirculation means. But in terms of amino acid and protein metabolism, we're just going to talk about how these amino acids are going to get taken up to the liver by the portal vein. So let's talk about transamination in a general way. Yes? So all of the catabolism that we're going to talk about of amino acids, the transamination and deamination, we're talking about it's happening in the periportal hepatocytes predominantly, right? And that's around the portal vein. I have a picture later that I'll show you. So you remember transamination from biochem? Maybe? No? I'm getting some shakes, so I will go back over it. So transamination is about taking one amino acid and one carbon skeleton, which is, I'm calling it carbon skeleton, but it is really technically called an alpha keto acid. Do you remember that? Alpha keto acid of another amino acid. So it's kind of two amino acids where you've got one in the actual form of an amino acid where it's got the amino group on it, and then the alpha keto acid of some other amino acid, and they switch places. I mean, the amino group switches. And so what happens is the amino group of this amino acid gets transferred to this, and often, I've written it as the most common partner in transamination reactions. It's going to be alpha ketoglutarate, which is the alpha keto acid of glutamate. Remember what I said about glutamate? What did I say about glutamate? What is its distinguishing uniqueness in life? Where does it rule? Intracellular, right? It's the intracellular hub, if you will, of amino acid metabolism. It's very important, most important intracellular amino acid is glutamate. At the same time, when I said that, I was calling out the importance of glutamine, its cousin, as the most important amino acid in circulation because of the fact that it carries amino nitrogen around. Okay, so glutamate and glutamine are very different. I mean, there are critical differences even though they're cousins. Glutamate is the intracellular hub of amino acid metabolism. It is the one that is present in a lot of cells, and right where we are right now in the periportal hepatocytes, it's critical. And so it's going to accept the amino group from this guy to become glutamate, right? The amino group is here now, and it leaves this alpha keto acid of some other amino acid. Okay, that's the general description. Note that transamination reactions are two-directional, bidirectional, right? They are reversible. That's what that's trying to show there. Okay, they are reversible. They're catalyzed by an enzyme class called aminotransferases, and they require this, PLP, pyridoxal phosphate. And that is not written there, so please, this is just so you can spell it correctly. Pyridoxal phosphate, so that's a cofactor. So anyone, I'm going to move to this one. Oh wait, did you get it? Everybody? Pyridoxal phosphate. Does anyone know what vitamin that's the active form of? Close. It is a B vitamin. It's not niacin. So B6, vitamin B6, it's one of those. And if you ever get to look at the biochemistry of it, it's a very cool way that vitamin B6 as pyridoxal phosphate gets involved in transferring the amino group. It actually takes the amino acid group and transfers it to the other alpha keto acid. Okay, so pyridoxal phosphate. The most common transamination reactions are AST and ALT, which I think I will show you at some point. Actually, I may not show you ALT. So, or yes, I do. I have them in sequence on both slides. So aspartate aminotransferase and alanine aminotransferase. Are there any questions about this? So this is, the key points are transamination reactions are always reversible, okay? And they always require vitamin B6 in the form of pyridoxal phosphate. So you need to know that it's vitamin B6 and you need to know what that stands for, pyridoxal phosphate. Now, I'm going to say this. In the fed state, you are unilaterally moving towards the production of glutamate. And I know it's break time, correct? 11:30? So make sure you write that down. We're moving in the formation and the direction of making glutamate, and there's good reason for it. We'll talk more about that when we come back from break. So take 10 minutes, be back at 40, please, 20 minutes till noon, and we'll see you soon. I think most people are back. Can we get started again? I think most people are back from their break. At least it looks like mostly you're back. So I want to say one more thing about AST and ALT. They are present in a lot of different tissues. Actually, they're very, very common, around the body. Certainly the liver has high concentrations of both of these transamination enzymes, and if you think about it, that's because they do so much with amino acid metabolism. Heart and muscle, these enzymes are highly prevalent as well. And the way we use these enzymes, we can use them clinically to help us differentially diagnose somebody, for example, with an MI. And it's not the only thing, of course. Nothing clinically is ever one piece of information. But we know that heart muscle tends to be much higher in AST than ALT, whereas liver tends to have more ALT than AST. And there may be a good reason for that we could talk about. But if you have had an MI, you tend to have really high elevations of AST and normal ALT. Now, why do you think that would happen after a myocardial infarction? You know what that is? So you're damaging your heart. Why would AST be elevated, do you suppose? Where would that, and that kind of gets to where transamination is going on. Kind of probably a rhetorical question that I didn't ask very well. So if you think about these enzymes, they are inside cells, right? So if you've damaged your heart muscle, you've broken cells open, right? So these enzymes, AST in particular, will be spilled into circulation. AST in the plasma or serum will be elevated after a heart attack. Those of you that are with me in MNT, we're going to talk about what happens in pancreatitis, where you have elevated other enzymes. So enzymes are intracellular, but they will get into circulation when tissue gets broken. Were you going to say something? You had your hand up before. Sorry, myocardial infarction. Okay, sorry. I have a bad habit of that, so don't let me do that. Okay, so that's just how these enzymes are really relevant to amino acid metabolism, but they're also used potentially clinically for various reasons. And I'm having trouble. First of all, make sure I'm recording. Why is this not moving? It's frozen. Isn't that nice? Pardon me for just a moment. You can talk amongst yourselves again for a second. May I get some help, Cheryl? Okay, let me stop recording. Yay! Okay, we're back. All right, so key points about transamination. This is kind of bottom line, what I want you to know about transamination. The first point, why is it important? One of the things that transamination does for us, it's a vitally important way of dealing with amino acid nitrogen to help us feed it into the urea cycle. So it's the first step in catabolism of most amino acids. And it is the way that we get amino nitrogen into the urea cycle. And we're going to talk about urea synthesis next, because that's one of the major points, and I'm sure you all have studied the urea synthetic pathway before, I bet, right, in biochem? Is that a, nah, okay, well, you'll know more about it after we get through. So, vitally important way of dealing with amino nitrogen for excretion of that nitrogen by urea cycle or by urea synthesis, okay? Number two is it's a very important way of equilibrating amino acid, nitrogen, and carbon. Basically, you can make, from one essential amino acid, you can make a non-essential amino acid, right? So we are able to transfer amino nitrogen around using transamination reactions so we can use the carbon skeleton from a an essential to make a non-essential and turn one non-essential into another non-essential. Remember that I'm not saying we can make an essential amino acid. We can't make the carbon skeleton. But we can make a non-essential from an essential. This is going to get confusing fast if I say this again. Please don't make me say it again. Hopefully that was clear. And then we can take one certain non-essential over here and make it into a different non-essential over here, right, with transamination. Is that clear? So it's a way of equilibrating amino nitrogen, keeping various amino acids available for tissues, right, because we need to make proteins out of them, so we need amino acids to be available. All right, which transaminases are the most active? They are the most abundant. AST and ALT, okay, know what they stand for. What are the three key amino acids that are involved in AST and okay, let's talk about it. AST, what are the amino acids that are required in AST? Aspartate and? Say it louder, will you? This room is terrible, so you have to really shout if you want to get heard. What's the other one? Glutamate, right? Glutamate is the major intracellular hub. So when in doubt, guess glutamate. No, I don't want you guessing. I would like you to just get that in transamination reactions, because you can guess when I ask about what's the major one in circulation, you're going to mess up because it's not glutamate. Glutamate is intracellular, critical for transamination reactions. So you're always going to have glutamate. With aspartate transaminase or aminotransferase reactions, you need aspartate with glutamate. What about alanine? So we know it's glutamate and alanine, right? It's in the name. So if you remember the name of the enzyme, so that's when you say ALT in your mind, you got to say alanine aminotransferase. Then you remember, oh yeah, it's alanine and glutamate. Okay. So those three key amino acids involved in AST and ALT, if I worded it that way, it's going to be glutamate, aspartate, alanine, right, for those two enzymes. Which amino acid? Okay, you know this one. Okay, all together. How do you abbreviate glutamate, by the way? GLU. Okay, great. These are reversible reactions. Which direction predominates in the fed state? You're producing what? Glutamate, right? And I've written them left to right like that. They are reversible. You need to know that. Remember that they are, but I've written them in the way that it's going to look in the fed state. You're going to be producing glutamate from the alpha keto acid of the other. Which vitamin is required? Right, as what form? Right. That's what I'm hearing. I know you know it. PLP, right? Pyridoxal phosphate. Just make sure you know what it stands for. Okay, here's a new question. Do all amino acids undergo transamination as the first step? I said most, right? Not all. So there are two key exceptions. And we're not going to go through. Yes? Oh, you know what? You're absolutely, well, it is on, but it's not picking me up. Sorry. Is it dead? It is dead. Whack, whack. No, sorry. Can I get your help? Is there a battery around here? I can shout. There might be batteries. Are these batteries? No, that's a photo lamp. What is this? Not anything. You gotta, I mean, that would be kind of fun. I have no idea. Shoot, Cheryl. Okay, so I just have to shout. Can you hear me? That's going to take some energy. Okay. So my general propensity is to go down, la, la, la. So if you please up there, start going like that, and I'll know that I'm not being loud enough. Does this work? No. No, the recording is fine. Okay. No batteries. No, oh, man, these rooms are so... If you'd be so kind, would you go up to the main office and see if you could get some batteries for me? Ask Sue Winkleman. Okay, all right, I can emote. I'm not the worst at that. Um, by the way, you know, that's something that you guys need to know too, right? So you know that you may very well be in a position where you have to emote. I was at a conference and we were in this room with this huge fan going, wah, wah, you know, and people kept talking and they would say something, they'd ask a question, and you know, no one could hear at the front. And anyway, I was called upon to shout for people. So I can do that. So the two that are highly, or that do not undergo transamination as the first step are lysine and threonine. And if you look in the Stepanek textbook, you'll see all sorts of interesting pathways for their catabolism, and I'm not going to ask you about them. They're highly convoluted. Both of them, at some point in their degradation, do have a transamination reaction way down the line. So the question is, do all amino acids undergo transamination as the first step? And the answer is no. Lysine and threonine do not undergo transamination as their first step. The other thing you need to know is that lysine is highly conserved. It's one of these amino acids that hangs around in circulation pretty long. It's one of those that just, for whatever reason, is not degraded very readily. And I'll say why the reason is. If you look at Stepanek, you'll see how convoluted the catabolism is, and you'll understand that lysine just avoids it because it's just too much trouble. I'm pretty sure. I have no idea really why that is, but anyway. And it's good that it is because it's an essential, so isn't it? Yes. I have to okay. What else? Another major point that I didn't put on this slide. Transaminases have a low affinity for their substrate. Is that right? Yes. Low affinity, high Km. You remember that? I vaguely, if you asked me to totally explain that, I probably would have a hard time exactly explaining that. Okay, but it's a low affinity or high Km means that you got to have a lot around before you have a lot of activity, right? Low affinity. So that's important. Can you think of why that's important? So under normal circumstances, in the post-absorptive, right, not fed, amino acid concentrations are going to be fairly low, right? What would happen if they had a high affinity, low Km? The liver especially, right? Well, and you certainly would be taking them all away from every other tissue that might need some access to amino acids, right? I mean, if you think about, you know, the job of the liver is to do what it does. It extracts a lot of them in the fed state, and in the fed state, you know, you're going to have high activity of those transamination reactions because you're kind of clearing. But then you're going to, at some point, you're going to reach an equilibrium, and you're going to let the rest of them go. And that's really nice, right, for the rest of the tissues in the body because they need access. We've got first pass metabolism going on, but the liver can't hog them all up, right? So we need to be able to release them. So low affinity, high Km for transaminases is a very nice feature of those. It's very important that those have that. Yes?
So is that for both of the substrates?
Yes. So the question was, is that for both directions? Yes. So you know, and these are, and we're not going to get more into the they're not highly regulated. They're constitutive types of reactions. They don't require energy, right? So they're going to go, when the substrate is high, they'll push it in one direction or the other. And in the fed state, you got a lot of amino acids coming in. They're going to be making glutamate out of alpha-ketoglutarate because that's predominantly what is happening in the paraportal hepatocytes. Gosh, how does it favor glutamate? It favors glutamate because you've got a lot of other amino acids. I mean, aspartate and alanine in particular are coming in high concentrations relative to glutamate. That's how it happens. Thank you, because I had to think about that. It's concentration driven. Okay? We're going to talk about what happens in the intestine. in the fed. So we're going to stay in the fed state, and I'm going to help you it'll be more clear. Alanine is one of the major products coming from the intestine from its metabolism of amino acids, what it does. Okay, so alanine is going to be higher than glutamate, and that'll drive it in that direction in the fed state. That's a great question, actually. Okay, any questions about any of that? All right, the second way I said, or actually Jonathan said, that we will catabolize amino acids is through deamination. Okay, and so here we go. Remember our exception? One of them anyway. Lysine is way out there, but threonine, the thing that it does is it undergoes deamination as the first step in its catabolism. So not transamination, but deamination. And it's a dehydration reaction. It also involves pyridoxal phosphate. And this is what it forms. Alpha-ketobutyrate is its carbon skeleton. Okay. What do I have up here? This is just a little tidbit for you that I have mostly. I'm going to just tell you that for the most part, I think I've written these reactions whenever we deaminate or we take an amino group anywhere. I've written it as ammonium. And that's because most of the ammonia that's in a cell is going to be in the form of ammonium ion, and that's because the pKa is about 9.5. So it's high. It's going to be in the plus state, okay? I may slip and just use NH3. It's the same thing. For our purposes, just think of it as ammonia. It's just, you know, what form it's in. So that's why I have that on that slide. All right. Now, let's go back. So transamination reactions, again, in the fed state, is going to be generating glutamate, pushing the reaction in the direction of glutamate. Then what, right? Why is that so important? So in the fed state, you're making glutamate, and the next thing that happens is that glutamate is going to get deaminated. So it's a transamination reaction that makes glutamate, with AST and ALT leading the pack, but there are other ones, okay? You make glutamate, then you deaminate glutamate by glutamate dehydrogenase. And that enzyme, okay, so you remember dehydrogenases, whenever you see those, you're going to be starting to think about reducing equivalents, right? Does that ring a bell? NADH being formed typically? Maybe FADH2? Do you remember that? Did you guys learn that yet in biochem? I'm seeing blank stares. It's hard in this room. Okay. So that'll form a reducing equivalent, NADH, and ammonia. So it's basically taken the amino group that came from some amino acid, it gets transaminated to make glutamate, and then glutamate, you get rid of that amino group by dehydration through this glutamate, I mean not dehydration, sorry, through glutamate dehydrogenase. I will abbreviate this in shorthand some places, GDH, glutamate dehydrogenase. That's a standard, right? You remember other ones like malate dehydrogenase, and right? Can I see a show of, or this is really foreign? Have you not had that yet? A little? PDH, thank you. Okay, so PDH. Maybe you haven't gotten to malate yet. Okay. All right. So we can call that first two-step in amino acid catabolism as transdeamination because it evolves, and I'm probably getting low again, aren't I? You probably, did you get it?
Yeah.
Yay! Everybody clap for Cheryl. Thank you. Yay, Cheryl! Thank you very much. Okay. Now we can go back to talking lazy. No, I'm just kidding. I will try not to do that. Can you hear me? Can you hear me? Hello? Hello? Hello, hello? No, it's not working. Okay, I'm going to power it off. Powered on. Test. Whoa, hello? It is working, so that's so funny. Okay, great. Can you hear me now? Okay. So transdeamination. And this is a funky way to write in a reaction. Hopefully you can gather this, that you've got an amino acid coming in from wherever, combines, gives its amino group to alpha-ketoglutarate to make glutamate, leaving its alpha-keto acid. That's the aminotransferase action. And then glutamate undergoes this glutamate dehydrogenase using NAD, but makes NADH. Remember how many ATP come from NADH? Remember that? 3, roughly. It's like 2.7 something. We'll say 3. And it also yields ammonia, which then feeds into the urea cycle. Now we're going to talk a little bit about cellular compartment. Transaminases are present in both the cytosol and mitochondria. Okay? Transaminases or aminotransferases are present in both the cytosol and the mitochondria. This is all happening, by the way, in the periportal hepatocytes. We haven't moved from that location. And glutamate dehydrogenase is a mitochondrial enzyme. So we've got a carrier that brings glutamate in to the mitochondria. And that's helpful. Glutamate, sorry, now I'm pulling on your ear here. Let's see if I can bring that down. Okay. So glutamate dehydrogenase and the first 2 steps of the urea synthetic pathway happen in the mitochondria of periportal hepatocytes. That's very nice. When you have the same location, you tend to have coordinate regulation of enzymes and you have more streamlined processes, okay? So the fact that glutamate dehydrogenase and the first 2 steps of urea synthesis happening in the mitochondria of periportal hepatocytes is an important point. So again, in the fed state, we're making glutamate. The next step, this enzyme, glutamate dehydrogenase, is responsible for producing a reducing equivalent, 3 ATP. That provides most of the energy required for urea synthesis. So it's highly coordinated, that this transdeamination is helping us generate the reducing equivalents that could be used to make urea. Because urea synthesis is very energy efficient. Can you think of why that might be important, that it's helpful, that it doesn't cost a ton of energy to make urea? Say that out loud. What is toxic? No, not. Ammonia. Ammonia is toxic, right, to the body. We need to be able to deal with this lots of amino acids, especially people following the Atkins diet, right? I mean, or Americans in general. Eat a heck of a lot of protein at a meal, right? We're breaking up a lot of it. What would happen if we couldn't efficiently deal with that by making urea? It would not be good, right? Ammonia is toxic. So very important that we can do that in an energy efficient way. Okay, so now that we've removed the amino, whoa, sorry. I always forget about that. And it was my now 16-year-old that probably did that, or Jillian, maybe the 13-year-old that, gosh, Mom, you got to use some animation. It was probably 2 years ago that I forgot about that. Okay. So how do we get rid of the ammonia we're going to talk about? And then we'll talk about what happens to the carbon skeletons, okay? So first of all, we're going to talk about the ammonia. Maybe while we're on this slide, because the next one is rather busy, let's talk about sources of ammonia in the body. Where is the ammonia coming from? So I'm going to help, we're just going to list them out, okay? So the first one we've already talked about. Ammonia comes from amino acids, right? When amino acids are deaminated, right? That ammonia, NH3, is released. So from deamination of amino acids. Number 2, ammonia comes from foods, actually. We get it in foods. Number 3, ammonia is released from bacteria acting on urea in the colon. We produce ammonia. It gets from the gut. Bacteria that have urease, the enzyme that works on urea. Not all bacteria do that, but there are a good number that do. Okay, so bacterial synthesis or release from urea. Number 4, nitrogenous compounds. Remember the building blocks of DNA and RNA, and we call those purines and pyrimidines. Do you remember that? Those bases. nitrogenous bases. Purines, P-U-R-I-N-E-S, and pyrimidines, P-Y-R-I-M-I-D-I-N-E-S, the building blocks of DNA and RNA. And then the last source is ammonia coming from the intestine. Metabolism there, we're going to talk about that. So a little trivia is about 1/4 of the urea that we make gets recycled by gut hydrolysis in the gut. Okay, yeah. Ammonia that is coming from the intestine, from amino acid metabolism there. Okay, we ready for the urea cycle? It's what you've been waiting for, I'm sure, to revisit the urea cycle. Urea synthetic pathway really is what we should call it, because not all of it is a cycle. This part's the cycle down here, okay? And I know this is a busy slide, so the next sequence of slides breaks it up into each of the steps for you. So I know you had to learn this in biochem, right? Did you get some of it? Did you have to learn the structures or anything? No, but you did have to kind of learn. Did you learn the enzymes? Yeah, okay, so hopefully this will be a review, but I'm going to go through it as if you don't remember anything, because I know how that is. I was sitting in your chair a long time ago. And I'm trying to think whether I really want to point anything out except for, I'm going to come back to the big picture in a minute, so I think I will just, oh no, I did it. Okay, so I've got it animated. So let me just, we'll walk through it, just so you, so remember that ammonia that just came from glutamate dehydrogenase, okay, as one source, right? And then there are other sources that ammonia is around from, but that's going to combine with CO2 to make carbamyl phosphate. This is a one-carbon entity. The enzymes, note that the enzymes are in yellow and purple, yellow boxes, purple writing, and just know that this takes energy. And I'm going to just click through and then we'll talk about each of the unique steps. The second enzyme, ornithine transcarbamylase, takes ornithine plus carbamyl phosphate to make citrulline. Step number 3 is this one where we have citrulline combining with aspartate. So citrulline plus aspartate goes to arginosuccinate. Step number 4, catalyzed by arginosuccinase, we end up yielding fumarate and arginine, and then the last step is we cleave arginine to yield urea. Okay, that's big picture. Now we're going to get into the nuances of each step, and we'll talk about regulation and various other things. Yeah, that's step number 8 is the recycling of fumarate. Okay, we're going to talk about each of these steps in turn. Okay, so step number 1. So important point, CPS1, I want you to know what that stands for because it helps you know what it makes. So don't just learn it as CPS1, learn it as carbamyl phosphate synthetase, because then you know, oh, it's making carbamyl phosphate, right? Very important point is that it cleaves 2 ATP equivalents up at the first step. The other thing that I want to point out is this NAG, okay? That's a terrible acronym, but anyway, stands for N-acetylglutamate. All right, N-acetylglutamate. This is what we call an allosteric activator of the enzyme. That means it has to bind tightly to CPS1 to make it work. So if you don't have N-acetylglutamate around, you will not get CPS1 activity, period. The other thing you need to know is that it requires magnesium also to stabilize the ATP cleavage and the ATP part of that reaction. So you remember that probably from biochem, that often when you see ATP, you're going to either see magnesium or manganese as cofactors to those reactions because they require those minerals to stabilize the ATP interaction with the enzyme. Does that ring a bell? Have you learned that level of detail there? What else? The carbon is coming from CO2. When you see bicarbonate, that is CO2 coming from metabolism of nutrients and amino acids and, you know, all of the ways that CO2 gets produced. So bicarbonate, that's CO2, comes from, that carbon is coming from CO2. And then you know where the, well, we'll talk about the ammonia. One of the nitrogens that is going to be in the final urea product down here comes from ammonia, okay? And I'm going to give you a summation about the urea cycle in a moment. And then remember I said the first two steps take place in the mitochondria of periportal cells, and so technically they're not part of the cycle, these first two steps, okay? They're part of the pathway of urea synthesis, but they're not technically cycle. Cycle means you're regenerating, right? So you always have to think about cycles, keep going, like the TCA cycle or whatever. And I think I have written out, yes, right here, so you're using 2 ATP, and this is the level of I'm going to ask you to remember is what steps is ATP cleaved. You need to be able to describe the energetics of the urea cycle, and we'll go over those as we move forward here, okay? So the first step uses 2 ATP. I've written this out for you just because this is an important part of regulation of CPS1. We're going to get back to that at the end after we've gone through all the steps. I'll go through the regulation of the cycle or the synthetic pathway. Acetylglutamate synthetase is the enzyme that makes N-acetylglutamate, right? And so its regulation drives how much NAG is around, and how much NAG is around drives CPS1. So it's important to know about both of these enzymes in relation to CPS1. Okay? Any questions about that so far? Okay, let's move on. Step number 2. Now we've got ornithine, which is 5 carbons, and carbamyl phosphate joining to make citrulline through this ornithine transcarbamylase. You can write OTC because I have abbreviated it where I've needed to because of space. These are really long names, but I want you to not just learn it as OTC, learn it as ornithine transcarbamylase because it'll help you know what the substrates are and how it's regulated is based on substrate and feedback and all that. We'll talk about that more in a moment. So step number 2, both of these in the mitochondria. One mitochondrion, many mitochondria. Okay, and then we have the cycle beginning with citrulline being transported out into the cytosol. It will combine with aspartate using this enzyme arginosuccinate synthetase to make arginosuccinate. And guess what? This is where another, how many ATP, right? It's written on your slide, I know, so I can't really ask that. But it's 2 ATP equivalents. So you remember when 1 ATP gets cleaved to an AMP, that's the same thing as 2 ATP? Can I see nods? Okay. Right, so remember that. That's where people get hung up. They think it's only 1 ATP. It's 2 ATP equivalents because you end up with AMP plus the pyrophosphate. So that's step number 3. So aspartate is where the second nitrogen comes from for urea. Think about aspartate. And I'm not showing you it's a disservice not to show you the structures, but it would be an incredible mess to show you the structures. I should have probably tried to figure out how to draw them in here. If you want to, the Stepanek book, I think, has the structure, so you can see that there's a nitrogen. I mean, you know aspartate has a nitrogen. These are just carbon entities. They don't have amino groups on them. So the nitrogen from aspartate gets transferred to citrulline to make arginosuccinate. Okay, so that structure has, the argino gives it away, that there's a nitrogen component of it. Step 4, arginosuccinate now gets cleaved to make arginine and fumarate. All right, and that is arginosuccinase that does that. Step 5 is arginine gets cleaved to yield urea and ornithine. And now, I don't have it on the same slide now, but ornithine goes into the mitochondria when citrulline goes out of the mitochondria, right? It's the same carrier. So this orange or this purple transport protein, ornithine and citrulline share it. One goes out while the other comes in, or vice versa. Now, let me think about what else I want to point out here. Wait, I forgot one more. I'm sorry, I got to go back 3 slides. Sorry, hopefully that's on the same page. Go back, sorry, not on the same page. The other piece around this, okay, that I didn't point out. Remember, it's ATP involving, so you need magnesium as a stabilizer. It's a cofactor. The thing about this step is it is the rate limiting step of urea, the urea cycle. It's the rate limiting step in the cycle. And it's where the cycle starts, if you have a beginning and an end to a cycle, right? We'll call it the beginning. It's the rate limiting step. Okay, now I'm going to click. The last, I think it's the last step that I have broken out here, is the recycling of fumarate, right? And so if you think about it, any cycle, you got to regenerate the entities so it keeps going, right? So fumarate gets converted to malate. Malate gets converted to oxaloacetate using that malate dehydrogenase. And I've kind of asked you before, but does that ring a bell at all that you have malate dehydrogenase? It's involved in the shuttle. There's a malate shuttle that probably you learned about. In so doing, it's also generating a reducing equivalent, which is important. And it makes oxaloacetate, which is really important because it helps us regenerate aspartate through the AST reaction. So what I want you to know in terms of fumarate recycling is that it, and I'm not worrying about fumarase here, I'm not going to ask you every single enzyme, but I want you to know malate dehydrogenase because it yields the immediate substrate for aspartate transaminase. So you need to know you need MDH and AST to recycle fumarate, okay? And the recycling of fumarate is really helpful because obviously that allows the cycle to continue. Are there any questions about the individual steps? And hopefully this is somewhat of a review and it's not just completely that you blocked it out of your mind so much that it's not familiar at all. Hopefully it's a little bit familiar. Okay, what next? Let's talk about the energetics, I think, is my next. So this NADH, again, produces ATP. So I had, don't be confused, really, when I talked about glutamate dehydrogenase also producing NADH, there's a fair amount of energy available, I guess is the point about urea synthesis, coming from NADH. This is a recycling that's produced that also, ensures that there's adequate energy around. So if you think about the net energy cost of making urea, how many ATP did we cleave? And there are 2 steps for that, right? So we had 4, right? 4 being used, and then we create 3, so the net energy cost is 1. Is that clear to everyone? Okay. So very energy efficient process. This is just written out for people that don't like cycles. I've just written out the steps so that it's linear, and some people like to learn it that way, so I'm not going to go back over it, kill you with that. But let me know if anything doesn't make sense based on what you've learned. Okay, where am I now? Okay, key points about the urea cycle. Let me go back because I like this slide better. All right, so this is the structure of urea, by the way. And so the first question is, for the urea, where does the carbon come from? Again, carbon dioxide, right? So CO2, and it's written as bicarb because that's the majority of it in a cell is going to be in the form of bicarbonate, okay? But it's CO2, carbon dioxide. where do the two nitrogens come from? And so I'm thinking, I want you to think about the cycle, the synthetic pathway. The first one comes from the first step, right? Comes from ammonia. And the second one comes from aspartate, where the rate limiting step is. Okay, so ammonia, one from aspartate. How much energy does it cost? Right, 1 ATP. Where does it happen? First of all, in the body. What kind of cell are we talking about? Periportal hepatocyte, so liver, but then specifically periportal hepatocytes. Where in the cell? So I want you to think about where in the cell of the, where in the periportal hepatocyte. First two steps, mitochondria, and then the rest in the cytosol. Fumarate and ornithine get recycled, and I want you to know how that happens. So you're going to know the pathway. The best way to do it is just write it out and understand what's happening at each of the steps. But it's important that those get recycled. Why? What would happen if we didn't recycle them? Do you suppose? You're thinking too specifically. What would happen to our ability to make urea if we weren't able to recycle those? I mean, you're thinking specifically, right? But generally, macro, a little more macro than that, Lena. Sorry? We're not going to make urea. We're going to stop it. I mean, it'll just not go. We won't have the cycle. So you need to recycle to keep that cranking. And it is cranking in the fed state, no doubt about it, because we're breaking down those amino acids because we can't use them all, right? At least until we get to a certain equilibrium, and then the liver lets the rest of them go. All except for the major group of essential amino acids, not major, but the one subgroup of essential amino acids that the liver doesn't really touch very much. And those are which ones are those that don't get broken down or transaminated and deaminated? And the branch chains, right? We'll talk about those when we get to muscle. So it's important that fumarate and ornithine get recycled so we can keep the synthesis of urea going. And how does it happen? I'm not going to go back over it. It's on those steps, okay? So I think you can try to take a look at that before next time and let me know if you have any questions. What regulates the urea cycle is where I want to go next. So any questions about anything so far? So let's talk about regulation. And so you're going to have to, I wish I could do both at the same time, is have the cycle up, okay? But if you can, have it marked where you can kind of flip back and look at it, it might be helpful to you if anything isn't clear from what I'm talking about here. So remember the first step, right? CPS1. Regulation is very important at that first step. What do you think might be the effect of having a high ammonia concentration on CPS1? And if you need to, flip back and just take a look at that reaction and let me know what that, I think you might be able to take a wild stab at this. High ammonia concentration. What's that going to do to CPS1, do you think? It's going to increase or decrease its activity? Decrease, I heard? Substrate, right? So it's going to increase. So, if you think about enzymes that don't look like, and it does have, it is regulated, it is highly regulated, but substrate-driven activity kind of holds for a lot of enzymes, right? So the more ammonia around, the more activity you're going to have with CPS1. What about NAG? What did I say about NAG? It's essential for it, right? So without it, you don't have any activity. So, it's critical for its activity, NAG. So you need NAG to allow, actually, so I said it's an allosteric activator. NAG allows ATP to bind to the enzyme, and without it you can't have any activity. So bottom line, CPS1 will not function. There's a third bit of regulation for this enzyme, and that is what controls NAG production. So remember I said the enzyme, I wrote it out for you, is acetylglutamate synthetase. That enzyme, its activity is controlled by, or is increased, its activity is increased by a high arginine concentration. Where is arginine in the urea synthetic pathway / cycle? Where does arginine show up? The beginning, the middle, the end? The end, right? It's the last step. Arginine gets cleaved to yield urea, right? So if you have a lot of arginine around, what might that tell the cell? I mean, why do you think that, and I'll just say this, let me back up a moment. So a high arginine concentration is going to increase the activity of the enzyme that makes NAG, N-acetylglutamate. It increases acetylglutamate synthetase activity. So we call that feed-forward activation from the end product of a pathway. If that starts to build up, or it increases in concentration, that's telling the cell there's a heck of a lot of amino acid there. The urea cycle's moving really quickly. I better replenish that allosteric activator that binds to ATP to the CPS1, right? So if it's bound up, you need to be able to synthesize more of it so that more CPS1s can get active. Does that make sense? So the end product, arginine concentration, as that increases, that's going to activate the enzyme, acetylglutamate synthetase, that makes NAG. And so then that'll ensure that CPS1 is going to be active in conditions when you want it to be. Okay. Next up, regulation at the ornithine transcarbamylase. So again, looking at the reaction, there's no ATP required. It's going to be really substrate driven and product inhibited. So you can know what the answer is here. What does high ornithine do? Excuse me? What's it going to do to OTC? Right? It's going to increase its activity because it's product. I mean it's the substrate, sorry. And then citrulline is the product. So if that's building up for whatever reason, it's going to feed back and inhibit the enzyme. Y'all with me? Okay. So those are easy ones. Now, this is a new point. Urea cycle enzymes and hepatic glutaminase, and I haven't told you about glutaminase yet, but these enzymes are coordinately regulated, meaning when the urea cycle enzymes are active, so is glutaminase, and when they're shut down, so is glutaminase in hepatic, in the liver. Okay, and I'm going to explain how that works and why it's important. Remind me again, Cheryl, when did we finish? 40? 35? 40? 40. 35. Why is it they can't just make it, forward, whatever, or something easy? Okay, 35, so we have about 7 minutes. So high protein intake and in general, fed state, right? When you're eating a, normal amount of food, normal diet or whatever, where you're taking in protein. You're going to increase activity of the urea cycle and glutaminase. Those are going to be active. They're going to both be hampered down in an extended starvation. Okay, they're both going to be less active, much less active in starvation. And why would that be? You will understand more in a moment when I talk about that. But it's important that they work together. Okay. Oh, what am I doing here? Here is. Why do I have this? Because I think I put this in as just to remind you again. Goodness gracious. Pardon me. I have extra slides in here. You didn't see that. I got to get rid of those. Okay. So let's think for just a minute. If you have a defect in any of the urea cycle enzymes, what do you think might what is the immediate result physiologically if you got a defect, meaning one of them's not working in this synthetic pathway? You mentioned it earlier. Not nitrogen, but ammonia, right? Right. So you're going to build up ammonia in circulation. If you can't, if one of these isn't working, then you're not going to be able to synthesize urea to any extent. So the question of, the direct metabolic consequence of this, metabolic and, I'm going to click, these are both asking pretty much the same question, right? What happens physiologically so you've got ammonia building up. What do you think happens? And many of you are not in MNT. We haven't even talked about it yet, but maybe you might imagine. So you've got a high amount of ammonia in the blood. What might you look like physiologically? You going to be doing well? Okay. There is something about that, but I think I want you to get a little, again, more macro, just in, you're going to be doing well? You're going to be walking around? It's toxic, right? It's toxic to the central nervous system, toxic to many tissues, toxic to the brain. So you will be in a coma. And people that have liver failure, that's the end stage of liver failure, is hepatic coma, because you've got buildup of ammonia. You've also got other toxic metabolites because of the failure of the liver. But a big part of it is that you've got toxic to the central nervous system. So coma and death are going to ensue if you can't make urea. And that's kind of the bottom line, what I want you to keep in mind. It's just a really important thing to think about. All right, the last thing I think we will get to today is just reminding you so we talked about where the nitrogen comes from for urea in a direct sense from the urea cycle, but how does amino acid nitrogen feed into the urea cycle? I'm sorry, I have to highlight this. So one is that we've got other amino acids that will be coming in via transamination and then feeding into alanine through alanine transaminase that will help us get to AST. So that's one way, is basically kind of feeding in. Alanine is a major way, but there are other amino acids that can transaminate with ALT, with alanine, to help get the amino acid nitrogen in to the pathway this way. And then over here by ammonia, where does the ammonia come from? So one of them we talked about already, glutamate, right? Glutamate dehydrogenase. And again, think about all of what we talked about, transdeamination, amino acid nitrogen equilibrating with alpha-ketoglutarate to make glutamate then undergoing dehydrogenation, the GDH reaction, right, to then yield ammonia. So that's one major way. And the other is glutamine. So I said that glutamine is a major way of transferring amino nitrogen around the body. So glutamine coming from other tissues will enter the periportal hepatocytes, and that's where glutaminase activity is very, very important to be coordinatedly regulated with the urea cycle enzymes. Okay? So glutaminase breaks down glutamine, leaves glutamate and ammonia, and then that glutamate can then be acted on by GDH to release the extra amino, right? So there are two amino groups in glutamine, and they both rapidly get taken off, first through glutaminase and then through glutamate dehydrogenase. So does that make sense that they would be coordinately regulated, urea cycle synthesis and glutaminase? And I'm going to show you more. We'll talk more about glutamine next time when we talk about the intestine and the other tissues. Okay, if there are any other questions, feel free to ask them now, but otherwise I'll let you let you get out of here. Yeah.
Is that at the bottom there, separate from the TCA cycle?
This is the urea synthetic pathway.
So with the fumarate and malate,
They are present in the TCA cycle, so it's not, this is paper chemistry, right? Biochemistry. So I'm writing them as separate pathways, but they are in concentrations in the cytosol and the, fumarate. I mean, they're around, right? It's always, you always have to write it as if it's completely separate and going on its own little,
they share the same.
yeah. Those enzyme machinery, the enzyme machinery is there.